So today I'll be talking about CHROMagar. CHROMagar, how to rapidly identify microorganisms using chromogenic media, right? So let's jump right into it. Chromogenic agar media are specialized culture media used in microbiology to isolate and identify specific microorganisms. And it is often based on the production of distinct, color changes. Yes, so this media essentially incorporates what we call chromogenic substrates. These chromogenic substrates, target enzymatic activities unique to particular microorganisms. You know that each microorganism produces unique, enzymes. So during the growth of this microorganism, these enzymes will react with the chromogenic substrates. And then when we talk about chromogenic substrates, they are basically molecules that release colored compounds when metabolized by certain enzymes. So the enzymes will react, with the chromogenic substrates to produce, unique colors. These unique colors are specific to what? The microorganisms. And this makes the whole identification process faster and more direct, right? So let's look at some of the key features of CHROMagar media. One, they are selective and differential. What they contain selective agent, for example, antibiotics or salts, which inhibit unwanted organisms. They are differential in the sense that the chromogenic substrates allow differentiations of organisms by color. Now, another thing that is unique to CHROMagar media is their rapid identification. Color development helps identify species or groups of microorganisms without additional biochemical tests. Yeah, so you can culture your sample on CHROMagar media, incubate for 18 to 24 hours, and thereafter you can directly identify the organism to even species level. So you don't need to go through, the biochemical, hurdles. Now, another thing that is unique to CHROMagar media is their common usage. It is used in clinical, in food, and water microbiology for detecting pathogens. So these are some of the features unique to what CHROMagar media. Now let's jump right into specifics. Let's look at some of the examples of chromogenic agar media. Just like we have nutrient agar, we have McConkey agar, we have saborodostress agar, we have what we call CHROMagar orientation. Now, CHROMagar orientation is used for urinary tract infection pathogens. CHROMagar orientation differentiates organisms like E. coli. E. coli appears pink, Klebsiella appears blue, and Enterococcus appears turquoise. Again, we have what we call CHROMagar candida. CHROMagar candida differentiates candida species. For example, Candida albicans appears green, Candida tropicalis appears blue, Candida krusei appears pink with rough surface. Now we have CHROMagar MRSA. It is selective for methicillin-resistant Staphylococcus aureus, that is MRSA, and colonies form mauve or purple pink. We have CHROMagar STEC. CHROMagar STEC detects Shiga toxin producing E. coli, that is STEC, and STEC strains appear mauve. Now we have what we call Brilliance UTI Clarity agar. It is for urinary tract pathogens. Distinct colors identify organisms like Proteus. Proteus will appear brown, E. coli as usual will appear pink, and a whole lot of them. Now we have what we call CHROMagar Salmonella. CHROMagar Salmonella is for isolating Salmonella species in clinical and food samples. Now, CHROMagar Salmonella produces mauve or pink colonies. And finally, we have Colorex VRE agar. Colorex VRE agar detects vancomycin-resistant enterococci. Now, colonies of Enterococcus faecius and Enterococcus faecalis appear in distinct colonies, right? Of course, we have many other, CHROMagar, we have CHROMagar staph aureus, we have, very many other examples of CHROMagar media. Now, let's look at some of the advantages of this CHROMagar media. We talk about they are faster, and more accurate, for the identifications of microorganisms. CHROMAgar media, simplify workflows by reducing additional testing. CHROMAgar media supports direct differentiation in mixed culture. Of course, CHROMAgar media come with limitations. Like you talk about the costs, they are more expensive compared to traditional culture media. And many of these CHROMAgar may require confirmatory tests. We use, of course, molecular identification is a gold standard we use in the laboratory. So it's not as if the CHROMAgar will take over, the molecular test. We need to go for molecular test for, which is the gold standard test for microbial identification. Of course, one of the limitations again is that they are limited to micros with available chromogenic substrate, right? So in summary, I've talked about CHROMAgar. CHROMAgar simplifies microbial identification using color-based reaction. CHROMAgar is useful across clinical and industrial microbiology, and it is a powerful tool. But like I said, it should complement, molecular identification and not to replace it. Molecular identification of microorganism is always the gold standard method for microbial identification. So thank you, thank you, thank you, thank you.